I'm Mr. Williamson, and I'm having a bad protein day. Hair made out of protein. That's what we're talking about today, protein and nucleic acids. Before we get started, make sure you're taking good notes. Cornell style works really well. Put it in your notebook, review them for the test. Let's get started. Proteins consist of 4 elements: hydrogen, oxygen, carbon, and a new one, nitrogen. All right. The monomer of a protein is called an amino acid. Sometimes they're called peptides. You'll hear about peptides once in a while, but most of the time we refer to them as amino acids. And I'm going to explain the structure of those right now. All right, there are 20 different amino acids. That's it, just 20 amino acids. All of the proteins that we have that are in existence are made out of those 20 amino acids. Think of it like the alphabet. We only have 26 letters in the alphabet, but all of our words come from those letters. It's just the combination and the order we put them in. So the difference between from and form, just the order. Same letters, just the order different. The same thing works in proteins. We take the amino acids, we put them in a different order, we put them in a specific order, and the protein does what it's supposed to do. So each amino acid consists of a central carbon atom, and I'm going to show you this, with a hydrogen atom attached to it, an amine group, a carboxyl group, and an R group. There is no such element as R. R is a placeholder. It's what makes the 20 amino acids different. So amino acid looks something like this. So as I said in the previous slide, an amino acid consists of a central atom, right there, a hydrogen atom attached to it, right there. Remember, carbon has to have 4 bonding sites. Show you all 4 of them. A carboxyl group, that's what this is over here, and you do not need to know the structure, all right? An amine group, and this is where our nitrogen comes into play. That's the new element, nitrogen. And then there's the R group. The R group is what makes this different, all right? If I showed you a different view, just a little bit, it's more accurate size of the atoms and things. But again, you have the carboxyl group down here, the central atom, the hydrogen, and the nitrate group. The R group is the thing that makes it different. There is no R element. So what am I talking about there? Well, these are the different amino acids. These are all 20 amino acids. No, you do not need to know these. Again, don't worry about it. They could be as simple as upper left, where it's just another hydrogen attached to it, okay, to the central atom, central carbon. Or it can be as complex as arginine down here, all right, where it has a whole bunch of, it's basically almost like a hydrocarbon chain. So each one of these is different. 20 amino acids is different by the R group. Whatever we attach to that central atom is what makes it different. But they all have 1 hydrogen, 1 carboxyl group, 1 nitrate group. It's the R group that makes it different. Every one of them. You don't have to worry about the structure or anything. You don't even have to worry about the remembering or anything, the form. Think of it this way, though. We talk about carbohydrates, they form rings. Lipids form long chains of hydrocarbons. I always thought of a protein kind of as a star. It goes out from that central atom, and then it comes down, and that's where the R group is. So I think of it as a star. So if you see a ring, you're probably talking about a carbohydrate. If you see a chain, you're probably talking about a lipid, and if you see a star-like thing, like what's on there, is probably an amino acid. All right, 2 amino acids bond together to form a dipeptide. And that process is done by, surprise, dehydration synthesis, removing the water from the combination of water, H2O, from each of the molecules, not water from each, but a combination to make water from each of the molecules we're combining to make one. Again, very similar to what we talked about when we made carbohydrates, all right, and to disaccharides or trisaccharides. Here's 1 amino acid over here, another amino acid over here. When we remove that OH, take that hydrogen out, we now can combine it into a dipeptide, all right, and that's dehydration synthesis. Now, looking at it, I can, do this. and we have a face because we recognize faces. It's an evolutionary skill. Seeing faces kept us surviving. Polypeptides are just long chains of amino acids. All right. Again, each time we attach an amino acid to the chain, it would be done through dehydration synthesis. The polypeptide starts out as like usual, amino acid. When we put them into a chain, like the second picture there, we have a polypeptide. And then this is way above anything you need to know. I'm just showing you how a protein would work. It's the way those form together, and finally we end up with a protein that is completely three-dimensional, all based on 20 different amino acids strung together in a chain, which all combines in a different forms because of the way they bond and things to make a three-dimensional protein that actually performs what it needs to be performing for the organism that's using it. All right, moving on. Let's talk about nucleic acids, complex organic molecules that store information in the cell. And for this unit, that's all you need to know. All right, you don't need to know anything else. All right, I'm going to explain a few things to you just to give you some background, but we'll talk about it later. Here we go. Basically, the monomer of a nucleic acid is called a nucleotide. All right. Nucleotides consist of a phosphate group, a 5-carbon sugar, and a ring-shaped nitrogenous base. Phosphate group, sugar, nitrogenous base. That's a nucleotide. What does that look like? Something like this. Here's your phosphate group right here. Here's your 5-carbon sugar. Here's your nitrogen group. That is a nucleotide. All right. Later on, you will know all these. And I'm so good that you're going to find it really easy to remember. All right. There are 2 types of nucleic acids: DNA, which you've probably heard of, and stored in the nucleus, and RNA, which is ribonucleic acid. DNA stands for deoxyribonucleic acid, and you can see underlined the thing there. All right. And ribonucleic acid, RNA. DNA is double-stranded. Probably should use a color you can see. All right. RNA is single strands. That's the big difference. That's it. So what do you need to know for the unit? What are nucleic acids? The monomer is a nucleotide. There's 2 of them that we have, DNA and RNA. They store information for the cell. That is the entire thing. All right. That's it. That's what we need to know for this. So let's take a quick review. All right. Proteins. They consist of the monomer is called an amino acid. An amino acid consists of a central carbon, a hydrogen, a carboxyl group, a nitrate, and an R group. It's that R group that makes the 20 different amino acids different. Probably should remember there's 20 amino acids. When you put them together, dehydration synthesis, removing water from the 2 molecules, making a peptide bond. Okay, dipeptide bond. That's what you need to know. Long strings of amino acids make a protein. Nucleic acids, a nitrate group, a phosphate group, a 5-carbon sugar, and a nitrate group. There are 2 different types of nucleic acids, DNA and RNA, and 1 unit, the monomer, is called a nucleotide. Hopefully you took good notes. Make sure you review them. If you have any questions, make sure you ask on an Edpuzzle or in class. Be glad to answer them. Good luck. Talk to you later.